Today I'm going to briefly discuss the difference between disinfection, sterilization, sanitization, and antisepsis. These terms are often used interchangeably, however, they do not mean the same thing. First off, it's important we control the microbial population in our environment in order to prevent the transmission of diseases and infection, stop unwanted decomposition and spoilage, and prevent unwanted microbial contamination on surfaces. Disinfection refers to the use of physical or chemical agents known as disinfectants, such as ultraviolet light, heat, alcohol, and bleach, to inhibit or destroy microorganisms, especially pathogens. Unlike sterilization, disinfection does not guarantee that all pathogens are eliminated. Disinfectants alone cannot inhibit endospores or some viruses, and the term disinfection is only used in reference to the treatment of inanimate objects and surfaces. Commonly, it's the use of phenolics, alcohols, aldehydes, or soaps on equipment or surfaces. So one example is using bleach to clean a counter, and another example is wiping down a stethoscope with an alcohol wipe between uses. Meanwhile, sterilization refers to the removal of or destruction of all microbes, including viruses and bacterial endospores, in or on an object. However, it's important to note that the term does not apply to infections proteins called prions, because standard sterilizing techniques do not destroy these. Sterilization indicates only the eradication of harmful microorganisms and viruses. Some innocuous microbes may still be present and viable in the environment that is actually still considered sterile. So the first example is a commercial sterilization of canned food, and the second example is preparation of microbiological culture media. Sanitization is the removal of pathogens from objects in order to meet public health standards. The first use is steam, high-pressure hot water, and scrubbing are used to sanitize restaurant utensils and dishes. Another example is chemicals are used to sanitize public toilets at a restaurant. So finally, antisepsis. The term aseptic describes an environment or procedure that is free of contamination by pathogens. Antisepsis is the reduction in the number of microorganisms and viruses, particularly potential pathogens on living tissues. So the first example is vegetables and fruit juices are available in aseptic packaging. And the second example is surgeons and laboratory technicians use aseptic techniques to avoid contaminating a surgical field or laboratory equipment. So I hope that brief explanation clarifies the differences between disinfection, sterilization, sanitization, and antisepsis. Thank you.